DNA replication is the process of using a DNA strand as a template for replication by adding nucleotides in their corresponding order to form two identical daughter strands of DNA. Replication begins at the origin of replication, where helicase unwinds the double helix at replication forks, separating the two parental strands by breaking the hydrogen bonds between the nucleotide base pairs and making them available as template strands. The replication bubble is the separation of the two strands at different locations on the double helix where replication occurs, so when the helix is unwound, the DNA appears to have bubbles where DNA replication is occurring. Molecules of single-stranded DNA binding proteins attach to the unwound template strands to stabilize them. A mutation is a permanent change to DNA. Mistakes can occur during DNA replication, but the cell has mechanisms to prevent and recognize errors in base pair matching, preventing mutations. There are enzymes dedicated to replacing incorrectly paired nucleotides if DNA polymerase does not catch the error upon adding the nucleotide. If a whole segment of DNA is damaged, nucleotide excision repair utilizes nuclease, a DNA cutting enzyme, to excise the damage. DNA polymerase then adds nucleotides to the resulting gap using the undamaged strand as a template. Because DNA polymerase can only add nucleotides to the 3' ends of a pre-existing DNA strand, the 5' ends of the daughter DNA strands are often incomplete, resulting in shorter and shorter DNA molecules after repeated rounds of replication. To protect the DNA from losing important genes, telomeres, consisting of multiple repetitions of one short nucleotide sequence, are located on the ends of the DNA molecules to protect the genes. In humans, the sequence TTAGGG is repeated 100 to 1,000 times. The shortening of telomeres contributes to cell aging, but telomerase catalyzes the lengthening of telomeres and restores DNA to its original length, compensating for the shortening that occurs during DNA replication. In normal human cells, telomerase does not play a large role, but with cancer cells, uncontrolled cell division and cell immortality can be attributed to telomerase.
Along one template strand, DNA polymerase III synthesizes a complementary strand continuously by elongating the new DNA in the 5' to 3' direction. DNA pol III moves towards the replication fork and pairs the nitrogenous bases. DNA pol III reads the template in the 3' to 5' direction, but it assembles the nucleotides in the 5' to 3' direction. Adenine is always paired with thymine, and cytosine is always paired with guanine. Because of DNA's structure, DNA polymerase can only add nucleotides to the 3' end, extending the duplicated DNA strand from 5' end to 3' end. Since DNA consists of two sugar phosphate backbones that run anti-parallel with the nucleotides arranged in opposite directions, the process of elongating the other new strand of DNA in the 5' to 3' direction is more complicated. DNA pol III must work in the direction away from the replication fork, creating the lagging strand because this new strand of DNA is not assembled in a continuous nature as is with the leading strand. The lagging strand is synthesized in sections known as Okazaki fragments. Each Okazaki fragment on the lagging strand must be primed separately with the primase, joining RNA nucleotides into a primer, marking the beginning of an Okazaki fragment. RNA primase synthesizes RNA primer, which is an initial nucleotide chain consisting of 5 to 10 RNA nucleotides. DNA pol III then adds DNA nucleotides starting from the primer, forming the Okazaki fragment. After reaching the next RNA primer to the right, DNA pol III detaches. This process continues along the DNA strand while DNA pol I replaces the RNA in the primer to DNA by adding nucleotides to the 3' end of the fragment. The second fragment is primed, and DNA pol III adds nucleotides, creating the second fragment, and detaches after reaching the next RNA primer. To connect all the Okazaki fragments, DNA ligase joins the sugar phosphate backbones of all the Okazaki fragments into a continuous strand of DNA.